So it's my pleasure to introduce Jan Vasbinder, who's well known for his endless efforts to bring together researchers across the globe to understand fundamental questions of science and society across and beyond disciplinary structures or boundaries. He currently leads the Paralimas Institute at the Nanyang Technological University in Singapore. And today, he will lead us in reflecting on the legacies of a towering figure in the field of complexity science, late Professor John Holland. Please join me to welcome John on stage.
On August 9, 2015, John Henry Holland passed away in Ann Arbor in Michigan. He had been a faculty member of the University of Michigan for more than 50 years. In an article that in the August 2015 issue of Communications of the ACM, Stephanie Forrest and Melanie Mitchell gave a beautiful overview of his legacy, his motivation, his focus, and his visions. And I strongly recommend that you read it. To quote from their introduction, John was a descendant of the cybernetics area. He was influenced by the works of John von Neumann, Norbert Wiener, W. Ross Ashby, and Alan Turing, all of whom viewed computation as a broad interdisciplinary experience. John thus became an early proponent of interdisciplinary approaches to computer science and an active evangelist of what is now called computational thinking, reaching out enthusiastically to psychologists, economists, physicists, linguists, philosophers, and pretty much anyone he came in contact with. End of quote. John was a great scientist, an explorer and a pioneer who established complexity as a new field of science. He was also blessed with an inexhaustible flow of original ideas. He once remarked, "I have more ideas than I can ever follow up on in a lifetime. So I never worried about someone stealing one of my ideas." But I will not talk about John, his science, or his ideas. I have known John for more than 30 years. So I would like to honor him by talking about that period and about John as a person and a friend and a supporter. The first time I met John was at a conference about artificial intelligence in Stanford in 1984. John talked about genetic algorithms. I did not understand much of his talk, and afterwards I looked him up and introduced myself. "Professor Holland," I said, "I am Jan and I'm from Holland. I did not understand a word of your talk." That was the start of an immediate friendship that deepened as time progressed and that has greatly influenced my life. It also led to the establishment of a European version of the Santa Fe Institute in the Netherlands, the Complexity Institute and Institute Paralimas at Nanyang Technological University in Singapore, and most recently the Complexity Hub in Vienna. Key in all this was John's infectious enthusiasm for and support of good and risky ideas. John was one of those unique people who can recognize good ideas in what others say and then make you believe you can do a lot more than you believe yourself. And then he supported you all the way when you were doing it, with other good ideas, encouragement, and with actions. At the end of our first meeting in Stanford, John invited me in Ann Arbor. There I got my first full exposure to genetic algorithms and to John's warm friendship. In the fall of 1988, John visited the Netherlands with his wife and daughter. My wife, two of my daughters, and I met them in Amsterdam. We had a great time, including dinner and a lot of giggling between the girls. But more relevant for today, John talked about something that he had gotten involved in and was very excited about. That was the Santa Fe Institute. One of the wonderful things of John is that you did not have to see him for a long time to deepen your friendship or continue your conversation. In fact, each time we met, we just continued where we left off the time before, even if there were years in between. In 1998, I visited John in Ann Arbor. My interest at the time was to develop new combinations of science, and I wanted to know what John thought about creating such combinations. John organized a dinner in his home with some of his friends and colleagues. And after dinner, all of us went upstairs to John's study. I forgot the topics we discussed there, but I will never forget the spirit of the discussion. It was like walking in a world of intellectual, fearless explorers. Later, John told me that this type of discussions took place in the Santa Fe Institute all the time. And it was always about some combination of sciences. That is when the idea emerged to start a similar institute in the Netherlands, or in Europe rather. Some years later, in 2003, the chief R&D officer of Akzo Nobel needed a keynote speaker for an in-house conference on innovation. I asked John. He came and stayed for a week. One evening we had dinner with a few leading Dutch scientists, and I had asked John to talk about the Santa Fe Institute, hoping he could convey his enthusiasm to a typical group of critical Dutch professors who asked questions like, if Santa Fe is so fantastic, why is there only one such place in the United States? John's answer was unforgettable. About 1,000+ senior scientists in the United States have Nobel Prize qualities, he said. That is the quality you need to make a place like Santa Fe work. That, and the capability to listen and be genuinely interested to find out what other disciplines have to offer. Only 10% of the best scientists have that capability. That limits the population from which Santa Fe can draw to about 100+ senior scientists, and the same is true for Europe. We seriously started our initiative to develop a European version of the Santa Fe Institute after that dinner. That initiative was energized by John's enthusiasm for the idea and his support in getting the right people to start with. John suggested that we should find some European Nobel laureates to endorse such a European Santa Fe Institute. That seemed like a great strategy, but not being a scientist myself, how do you get access to those people and how do you get them to support such an idea? No problem, John said. I will call Manfred Eigen and get him to support you. And so it happened. Manfred called 15 top scientists, 5 of whom were Nobel laureates as well. All the scientists Manfred called became founding fathers of the European Institute Paralimes. And one of those scientists was Bertel Andersen, at that time the CEO of the European Science Foundation. We founded Institute Paralimes on 31st of August 2005 in a castle in Ruurlo in the east of the Netherlands. John was there, and so was Brian Arthur, who had called a few days before, saying that he wanted to be involved. I asked John to talk about Santa Fe during the dinner before the meeting to excite the participants with the spirit of Santa Fe. John was willing, but suggested that Brian was a much better speaker, so you had better ask him, he said. Brian spoke for about 45 minutes, and the echoes of that improvised but highly inspiring speech were captured the next day in the manifesto that laid out the principles of Institute Paralimes. John again played a crucial role in getting the wording of the manifesto right, so that the Institute could do the explorative science that John believed was the key to Santa Fe's success. John could not attend the meeting in Strasbourg in March 2006 to formally start Institute Paralimes, but Brian, Manfred, and many others did. But John was present at the workshop in 2007 on conceptual neuroscience chaired by Jean-Pierre Changeux. Remarkably, John was the only speaker amongst the group of renowned neuroscientists who actually proposed a conceptual framework to study the working of the brain. And John was the keynote speaker at our first conference, Science Without Boundaries, in October 2007. At that time, John was already working with Helena Gao-Hong on language and complexity. At his suggestion, I met Helena in Singapore to ask her to speak at the conference in the Netherlands. During that meeting, Helena mentioned that John would be 80 years in 2009. That triggered the idea for a conference to honor John, to be organized by Institut Paralimes, the Santa Fe Institute, and to be hosted by the Nanyang Technological University. Bertel Anderson, who had at the meantime become provost of NTU, agreed, and the conference Adaptation, Order and Emergence took place in February 2009. We invited speakers in the field of interdisciplinary science to speak. They all came. That conference had far-reaching consequences. It spread the buck of interdisciplinary science to NTU. Subsequent meetings between the management of the Nanyang Technological University, John, and many others led to the decision to develop a complexity program at NTU. That program started in August 2011, and I had the privilege to be its first director. Its first target was to develop the Complexity Institute. That Complexity Institute was established in April 2014, and the two co-directors, Peter Sloot and Steve Lansing, have been organizing this conference as well. John became one of the members of the advisory board. Now you must know that the complexity program started, when the complexity program started, only a few people outside the university or inside the university had any idea what complexity was about. It was a kind of a magic term that gave a name to a problem and then allowed you to move on as if the world was simple. 3 years later, when the Complexity Institute was launched, Peter Sloot and Steve Lansing being their directors, that was a totally different situation. John had played a major part in making that difference, partly because of who he was, always listening carefully, always sharing his ideas, always on the lookout for new and interesting questions, and always without pretensions. Partly because he took active part in many of the missionary meetings we organized, and partly because of the one-on-one meetings he had with key people in Singapore and with the students in NTU. John put his heart in those meetings. He was genuinely interested in the other, and he did not push others to be interested in him. That is, I think, why he was so enormously appreciated and highly respected in Singapore. That is also, I think, why his message carried so much weight, and why he could have such influence in making the idea of complexity research accepted in Singapore. Given the Singaporean scene, that was quite remarkable. Without going into any detail, I should point out that in Singapore, science and research must always serve a purpose that is encapsulated in the answer to the question, what is in it for Singapore? Unlike many other scientists that visit Singapore, John never questioned this basic principle. In fact, in many discussions with Peter Ho and other Singaporean leaders, he always was on the lookout for ways in which he could help the Singaporean government to do a better job. John was greatly impressed by the way in which Peter Ho's group had developed risk assessment and horizon scanning as a way to get a grasp on the future. In one of his last emails he sent me, he wrote, "I'm planning to come to the meeting in March. I have a computer model of how RAS, the risk assessment and horizon scanning, works that I'm quite proud of." He had in fact arranged a meeting with Peter Ho. It was not to be. During his last visit in September 2014, we organized a meeting for John with the students of NTU. It was a 3-hour meeting in which John discussed some of his ideas for explorative research with the students. He was already very ill at the time, but it was John at his best. Peter Ho came to pay his respect. Some years ago, in a reflective moment, I thought about the people that had the biggest influence on my life. John is in the top. John changed my life, and John changed a lot of lives. John established complexity science, or complexity as a science, and John changed science. John changed government agencies, and in doing it all, John was a great friend to many people. I feel myself very privileged to be one of them. Thank you. Oh, thank you.
It's my pleasure to introduce our next speaker, Ed Bullmore, who is in the psychiatry department at the University of Cambridge. He's going to share with us his understanding of how the brain works and how that helps in the treatment of mental disorders.
Okay, well, good afternoon. I hope you can hear me okay. Can you hear me now? A little better? Can you hear me? Yeah, good. Well, thank you to the organizers for inviting me to contribute to this meeting. I am going to talk a little bit about connectomics in health and disease, with a focus on how we can use graph theory as a key tool to understand brain network organization. These are my disclosures, just so you know. I work half-time for the University of Cambridge and I work half-time for GlaxoSmithKline. Nothing I'm going to say today has any commercial sensitivity, but it's important for us to be clear about that. So what is connectomics? Well, it's a word that really didn't exist until quite recently. It's about 5 or 6 years old, the word. And as you can see from the slide at the bottom, this is an area that is growing with enormous speed. So if you look back to about 2005, the work in the neuroscience literature that used graph theory to understand brain networks was ticking along at a very low rate, bit of a niche interest, and then over the last 10 years there's been truly exponential growth, both in the number of publications and in the number of citations. So in neuroscience, this is a field that has sprung up pretty much out of nothing and is growing at a tremendous rate over the last decade. Why is that? What is driving the growth in this new field of connectomics? Is it just a kind of fashionable blip, or is it something more fundamentally driven by changes in science? Well, I will try and persuade you that this is not just a fashionable fad, that this is an important opportunity to think about how the brain is organized that depends really on two converging factors. First, we have the work represented by this community and by others that over the last 10, 15 years has seen the physical sciences grapple with the complexity of real-life complex systems across a range of different areas, from infrastructural networks like the global airline network, metabolic networks, gene-gene interaction networks, economies, ecosystems. There is a tremendous proliferation of complexity science, of network science in general, and that has provided new tools, new ways of quantifying the complexity of network organization. And the other trend that's important is more in neuroscience itself. We have seen the growth of new techniques for visualizing nervous systems. This image is a magnetic resonance image, an MRI data set of the human brain. This is using a technique called diffusion tensor imaging, which allows us to reconstruct with some accuracy the white matter tracts, the bundles of cabling, if you will, that interconnect areas of cortex in the human brain. And we have many other MRI techniques that we can use in humans and other large-brained animals to look at the macro-scale organization of the nervous system. There has also been tremendous recent growth in techniques that allow us to look at nervous systems at a finer-grained spatial resolution, so-called mesoscopic or indeed microscopic network organization. So it's these two trends, in my opinion, are the tools for network science, the tools for grappling with the complexity of a system like the brain, combined with new technologies out of neuroscience that allow us to visualize nervous systems across a range of different species and scales with unprecedented precision. Now, I'm going to give you the kind of gestalt view of what this means when we are talking about a human brain network. How do we get from an image of the human brain, which is what you may be able to see here. I wonder, incidentally, if it's possible just to dim the lights a little bit, just in front of the the screen just to make that a bit easier to see. So hopefully you can now see the grayscale background of the human brain, and on its surface, marked in color, are a number of different areas, which will become nodes of the network. So what we need to do to turn an image into a network is, first of all, we need to define where those nodes are, and then we need to draw connections between nodes based on their association with each other, and that could be their functional connectivity or their anatomical connectivity. And then we've rendered the nervous system as a graph, and we can play around with it. We can look at it in a topological space. We can look at the degree of each node, its modular affiliation, and so on. All of the tools of network science can be applied once we've reduced the image to a graph. Let me talk through that a little bit more detail. So we're starting here with a human brain in the scanner. And as I've said already, the first step is to define the nodes that will form a network. We talk about that as a process of parcellation. We're going to simply subdivide usually the cortex of the brain, the gray matter that lies on the surface of the brain. We're going to divide that area into a number of different parcels or nodes. And then if you had a functional MRI scan as the basis for your analysis, from each of those nodes you'd have a recording of physiological activity, low frequency oscillations recorded by fMRI while the subject is lying quietly in the scanner at rest. You can make those recordings from each node, and for a pair of nodes you can correlate their activity over time. So you can render the association between these two nodes as a correlation coefficient or mutual information or some other measure of association between the time series activity between each pair of nodes. We can repeat that analysis for all possible pairs of nodes, and typically we might have 300 or 400 comprising the parcellation template, and that produces this so-called functional connectivity matrix or association matrix, which shows us where in the brain there is synchronized activity over time. If you have a lot of structural MRI scans, you can do a similar kind of analysis. You can look, for example, at the thickness of cortex in each cortical area, and over subjects you can correlate the thickness in cortex between areas and produce a matrix like this, which summarizes the anatomical connectivity or structural covariance. If you have data repeated over time, you may be able to measure developmental change in the cortex, and I'll come back to this later. In adolescence, over the course of the normal adolescent period from about 14 to 24 years old, the cortex shrinks, becomes a little bit thinner, and that rate of shrinkage can be estimated at each region, and then we can correlate between regions based on the similarities that they show in that normal developmental trajectory, which gives us another kind of association matrix, this one sensitive to developmental change in the brain. So you can see there are a number of different ways we can get to an association matrix that summarizes the interdependencies in terms of structure or function or development between brain areas. And having done that, we can, if we wish, simply threshold the matrix, binarize it to produce the substrate for an analysis of brain network topology as an undirected, unweighted graph, which is, of course, the simplest possible graph theoretical model. So that is the process in a nutshell. It is a fairly simple analytic pipeline at high level. It is adaptable to a range of different human imaging technologies. I'm going to talk mostly about MRI today, but it's generalizable also to electroencephalography and magnetoencephalography, and a number of different other neuroscience techniques that we can't necessarily apply in humans. So it's generalizable, and it renders the brain, whatever the source of the data, in the same mathematical framework, allowing us, for example, very easily and naturally to look at the correspondence between network topology based on structural measurements such as DTI, comparing those to functional measurements from functional MRI or MEG. The key questions are: how do you define the nodes? How do you define the edges? That is work in progress within the field. But already, I think, enough has been done to suggest that this is an interesting way of looking at the brain and capable of generating quite consistent findings, regardless of minor methodological differences between groups. So if you look at what's been reported in the human connectomic literature so far, a number of key findings have recurred over and over again. First of all, it is clear that brain networks, human brain networks, have hubs. They have higher degree nodes, and you can see that represented here in terms of the degree distribution. That with degree on the x-axis and cumulative density on the y, shows us that there is a scaling regime for the degree distribution in humans and an exponentially truncated cutoff. So this is a degree distribution that belongs to the broad class of broad-scale degree distributions. It's not a pure power law, but it has some regime of power law scaling. You can map the location of those high-degree hubs anatomically, and you can explore the interconnectivity between hubs. And this has led to the discovery in human brain networks of a rich club, the phenomenon I'm sure you're familiar with, whereby the hubs of a complex network may be more densely interconnected to each other than expected by chance. In the human brain network, there is a rich club. There are also, by definition, some less well-connected nodes in the periphery, and there are so-called feeder connections that link the rich club to the periphery. That's one way of looking at the community structure. There's also been a lot of work looking at modular decomposition, and this is a topological layout showing the modular community structure of a human fMRI network. The nodes are differently colored according to their modular affiliation. You can see, as you'd expect, that there is dense intramodular connectivity and relatively sparse connectivity between modules, and most of that intermodular connectivity is mediated by a few so-called connector hubs, shown here as square markers. So these are some aspects of human brain network organization that have emerged recently and already been highly replicated. Now, what do we make of this? How do you validate or make sense of a completely new way of thinking about nervous systems that comes along as graph theory has done and begins to talk about topology, whereas for most of the history of neuroscience, we've really been very focused on anatomy. How can we begin to, as it were, domesticate these new metrics? Well, one way that we've begun to think about this is to look at the relationships in brains between their topology and their cost. Now, the brain is overall an expensive organ. It comprises about 2% of body mass in humans, but about 20% of the body's metabolic budget. So this is a very expensive piece of equipment we have in our heads. And if you look at how the cost of a brain is related to its topology, a number of interesting findings emerge. First of all, we can think about cost in terms of the length of wiring, the so-called wiring cost that links nodes in the network. And if you look at the distribution of connection distance in the DTI network, human DTI network shown here as a histogram, you can see it skewed to the left, meaning that most of the connections are relatively short. The brain is to some extent parsimonious and tries to keep its wiring cost down, but there is a tail of long-distance connections. Overall, the system is less expensive than a random graph, but not minimally expensive. If we look at the relationship between distance and degree, you can see there's a strong positive correlation. So the higher degree hubs tend to make more long-distance connections, and since we know that the length of connectivity is related to the cost of connectivity, that's one piece of evidence to suggest that some aspects of brain network topology may be more expensive than others. Of course, we can also look at the cost of the brain in terms of its metabolic demands, and MRI doesn't actually allow us very directly to measure brain metabolism, but we can combine MRI with another technique called PET, or positron emission tomography, which allows us to make measurements of glucose metabolism in the brain. And then we can see that the degree of nodes in an fMRI network is anatomically coincident with the locations of the most metabolically expensive areas of the cortex. And indeed, degree and metabolic rate are highly related on that log-log scale, again suggesting that the more high-degree elements of the connectome are more expensive, not just in terms of wiring, but also in terms of local metabolic demands. This is a a slide you've already seen in part. This is the Rich Club in the human DTI connectome, but the paper that introduced this concept also went a little bit further than simply describing the existence of a Rich Club in the human brain. They pointed out that this was expensive in terms of its connectivity. So if you look at the distribution of long-distance edges in the human brain, you can see that a disproportionate number of a disproportionate amount of the total connection cost, the total wiring cost of the human connectome, is allocated to connectivity between Rich Club nodes or connections from the pore periphery, the feeder connections from the periphery to the Rich Club. So this is expensive, and here is a hint of why that expensive aspect of topology might be worth it for the brain. If we do a path motif analysis in the DTI network, it turns out that the path motif that links a peripheral node via feeder connection to the Rich Club, through the Rich Club, and then back out via another feeder connection to a peripheral node, that motif is very highly overrepresented in brain networks compared to random, shown here, suggesting that the extra wiring cost of the Rich Club might be worth it because it facilitates communication between nodes that would otherwise be separated by a long topological distance in the network. One of the important and I think really powerful things about graph theoretical analysis in neuroscience is its generalizability. And that allows us to look across a range of different species and scales and modalities of neuroscience data to begin to ask questions, for example, such as how general are these principles of network organization? And here we went from the human DTI network, which is obviously a macro-scale description of one of the bigger brains in the animal kingdom, down to a microscopic organization in one of the smaller organisms, the famous nematode worm, C. elegans, which is about 1 millimeter long and has the distinction still of being the only organism that has been completely mapped in terms of its connectivity at a cellular scale. So we know that there are about 302 neurons in C. elegans and about 6,000 synaptic connections. It is a more or less complete wiring diagram of a complete nervous system at cellular or synaptic scale. If you apply the same Rich Club algorithm to this nervous system, it turns out that there is indeed a Rich Club in C. elegans. It is comprised of about 11 neurons. You can see they're located at the head and the tail of the animal. This is a topological representation of their interconnectivity. We obtained this result blind, more or less, to the biology of the worm, but when we took these results to a worm expert for interpretation, he was not surprised at what we'd found. These neurons that constitute the Rich Club, it turns out, are the command interneurons of the C. elegans that had previously been identified as critically important for coordinated movement of the animal. This topological feature shares something else in common with the human DTI network. It's expensive in terms of its connection cost. So if you look at the distribution of wiring cost or connection distance in C. elegans, most of the connections are short, but some are long, and most of those long-distance connections are related to the Rich Club. This same path motif that links to peripheral nodes via the Rich Club is also highly overrepresented in C. elegans compared to a random graph. So I hope you can begin to see that DTI networks have Rich Clubs that are expensive, but maybe facilitating in terms of short path length connection between peripheral elements of the network, and that exact same profile is characteristic also of C. elegans. The idea that emerges from these and other data that there is some kind of trade-off between connection cost and topology, we can explore not just descriptively, but computationally. We can use quite simple generative models to reproduce the organization of human brain networks and endeavor to learn something about the generative principles or selection pressures that might have led to the formation of brain networks. So this is a human fMRI network. We're just looking at the right side of the brain face on. So this is the external lateral surface of the cortex. This is the medial surface. The nodes are scaled in accordance to their degree. And we tried to reproduce that pattern using a number of generative models. This is one that simply penalizes the connection distance between nodes. This is one that trades off distance against a preferential attachment type term. And this one trades off distance against clustering. And in these data, we found that this particular trade-off between cost and topology, where the probability of a connection forming is a product of the distance between nodes, but also the tendency for nodes to share nearest neighbors in common. If a connection can form that links two nodes that have a lot of nearest neighbors in common, its probability will be increased even if they are a long distance apart. This is an example of a fairly simple two-parameter generative model that does a pretty good job of reproducing the complex statistics of human brain network organization. And it does so on the basis that penalization of cost is not sufficient. There must be a trade-off with topology. And that line of thinking has resonance also in other areas of network science, particularly computer chip design, where people are endeavoring often to put topologically complex designs onto a very constrained spatial embedding. And in that literature has emerged the concept of Rent's rule, in other words, the fractal scaling between the size of an area in the network and the number of connections to nodes in that area. This was originally observed in very large-scale integrated circuits. It's observable also in human MRI data. It suggests, again, that human brain networks may have formed as a result of competitive pressures between the functional advantages of a high-dimensional network topology and the spatial constraints of embedding that topology in the 3 dimensions of the head. Incidentally, knowing the fractal or Rentian scaling of the human brain, it's possible to derive the allometric relationship between gray and white matter that has been observed over a range of different mammalian species, again suggesting that these are quite important general principles and not just specific to human MRI. Now, I want to touch briefly on a new kind of data that's emerged and that may be of interest to people in the audience who are thinking about how to get started in brain network science, and that is track tracing data in the mouse. And this is important because it is in many ways the gold standard for the anatomical wiring diagram of a big brain, a mammalian brain. And the technique involves injecting a tracer in one part of the brain and then leaving the animal for a few days. That tracer will then propagate through the axons, through the white matter cables that link this brain area, the source region, to all other regions in the brain. The animal's sacrificed, and then the location of the tracer is examined microscopically. And you can see you get these very beautiful maps showing how the tracer is propagated here from the site of its injection along this curvy linear tract to a target region here. From these data, we can estimate the connectivity weight or strength of connection between any two regions. We can estimate quite precisely the axonal distance. We can even estimate the cross-sectional area of an axonal tract that links two regions and combine distance and cross-sectional area or bandwidth to give us an estimate of axonal wiring cost. Importantly, this data is asymmetric. It looks symmetric, the matrix, but it's not in detail. And it's weighted. The results I've shown you so far were based on binary graphs. Once we have data of this quality, where the range of connection weights extends over 5 or 6 orders of magnitude, we can apply more sophisticated methods of weighted and directed graph analysis. And when we do that, we discover things that in many ways we already knew. The mouse connectome has a modular organization. We can break down its community structure into anatomically co-localized regions that tend also to be functionally specialized. Again, of course, the algorithm knows nothing about this prior knowledge, but for those of you with a bit of neuroscience, you may be reassured to discover that the connectome of the mouse has modules that are, include regions specialized for particular sensory functions, motor functions, and so on. This is its community structure and matrix representation, highlighting that there are a number of nodes colored here in red that are not particularly well affiliated to any single module or submodule, but may play a particularly important role in mediating connectivity between modules. These nodes have a high participation coefficient and are important, as that term defines it, in mediating the connectivity between these more functionally specialized and anatomically co-localized modules and submodules shown here around the matrix diagonal. We can look again at those trade-offs between participation and cost in the mouse. The nodes that are most important in mediating intermodular connectivity have higher wiring cost. Those regions of the brain that have high participation are also enriched for genes that are functionally important for these higher order cognitive functions like learning, behavior, and cognition. And if we try and reproduce or regenerate the mouse connectome shown here using generative models that simply minimize connection distance, we can do a pretty good job of recapitulating the modular organization of the brain, but we cannot reproduce the high participation nodes that link modules over longer distances unless we allow our generative model to be adaptive to topology as well as connection cost. Another example, I think, of how generative models as well as simple empirical descriptions point towards the general idea of a trade-off between the cost of wiring a brain and its topological organization. I mentioned in the previous slide genes, and again, I think this is a very important sort of edge for the field right now. Most of what has happened in connectomics has been at the level of, perhaps necessarily, at the level of systems. And if I talk to neuroscientists about it, often what they want is some anchoring of that systems level analysis in more reductionist, molecular, genetic terms that they can understand as fundamentally biological. So it has been important, I think, in recent years that thanks in large part to the Allen Institute of Brain Sciences, it has been possible to link gene expression measured in the mouse and in the human to the topological organization of the connectome in the mouse and the human. And this is one example from recent work on the mouse. So this is another representation of the mouse connectome generated by track tracing experiments, as I've already described. And here is a parallel matrix. The rows and columns correspond to the same cortical areas as the nodes of the connectome, but here the measurements are not of track tracing, but of local gene expression. And so over all of the 213 regions in the mouse brain, we have about 17,000 genes that have been measured locally. And what the investigators did here was to look at the connectome, to look at its topology, and to ask the question, how does the strength of anatomical connectivity between nodes relate to the strength of co-expression of genes by those nodes? So here, for example, they have two nodes that are connected, and they calculate the correlation between the gene expression profiles for those two nodes. And what this illustrated was a couple of things, that the nodes, the edges that linked high-degree hubs and were therefore part of the rich club in the mouse connectome also tended to share genes, had co-expression profiles in common, and the genes that were particularly enriched in common between high-degree nodes that were interconnected anatomically were enriched for metabolic functions. So this is a new piece of evidence, a new kind of evidence indeed, to suggest again that the topological organization of the mouse connectome in this case is related to its metabolic cost, and that high-degree hubs and the connections between them may be associated with overexpression of the metabolic genes that are important for generating energy in the brain. And we can apply a very similar kind of logic, and have done so recently, to analysis of human fMRI networks. So this again is leveraging the data on human brain gene expression that have been released by the Allen Institute of Brain Sciences. We've been able to map those local brain expression changes to an analysis of the connectome derived from resting state fMRI data. And what we found in this experiment was that these areas, shown here as dark, large red circles, had a combination of longer connection distance and more intermodular connectivity. They were, if you will, the expensive connector hubs of the fMRI connectome. And those nodes specifically were enriched for genes associated with metabolic function, oxidative metabolic function. So this is a completely different species. Obviously, we're talking now about human. Previously, it was the mouse. But in both cases, you can begin to see how gene expression can be related to topology. And in both cases, the more expensive components of the connectome, the intermodular hubs with long connection distance in this case, are associated with enrichment of genes for generation of metabolic energy. So overall, I would say that there has been tremendous growth in the field, not just down here in the human, not just up here in the C. elegans or in the mouse, but across a range of different species by various different techniques. The complex topology of all of these nervous systems has been elucidated or is being elucidated, and there is emergent evidence for the general principle of an economical trade-off between how much it costs to wire a component to the connectome and its value to the organism. Now, I've slightly danced over the question of why it might be worth it for the brain to pay an extra price in terms of wiring or metabolic energy for particular aspects of its topology. This slide gives you a part of the answer to that question. If we look at what happens to the global efficiency of a brain network under random attack, shown here in blue, we can see it's pretty resilient. If we attack the nodes in terms in decreasing order of degrees, so we attack the high degree hubs first in a targeted attack, you can see global efficiency falls off much faster. Brain networks are resilient to random attack, but quite vulnerable to a targeted attack on their hubs, and that will degrade their global efficiency quite rapidly. Here we have two independent studies, both showing that path length, which of course is inversely related to efficiency, shown on the y-axis here, is inversely correlated with IQ. So higher IQ individuals here in a DTI network, here in an fMRI network, tend to have shorter characteristic path length, higher global efficiency. They're more integrated networks, and the in silico study shows that efficiency, that short characteristic path length, will depend on the hubs. So there's a piece of evidence joined up from two prior studies to suggest that intelligence, as one particular aspect of cognitive function in humans, depends on hubs that can mediate short distance connections between nodes. If you want more detailed analysis of how human network topology relates to cognition, the field at the moment is a little bit thin in this area, and there's no fundamental reason why that should be. It just so happens that most of the people in neuroscience who've been particularly interested in connectomics have tended to use anatomical data or resting state data, and of course one of the limitations of that approach is we know very little about the cognitive state of individuals while those data were being collected. But we can think a little bit differently about how to construct a network, and here is an example of such an approach. We used meta-analysis in this case to draw together published data on something like 1,500 primary task-related studies that had already been published and reported coordinates where brain tissues showed a task-related increase in signal or activation. And we compiled all those data, and we reasoned that if two brain areas were very frequently co-activated across a range of different tasks, we should draw an edge between those nodes to form a co-activation network which ended up looking like this. You can see, perhaps at a glance, that this has a lot of complex topological features in common with some of the connectomes I've shown you previously. It has hubs, a rich club, modules, and so on. But importantly, because the edges here are defined on the basis of task-related data, cognitively informed experiments, we can deduce something about the cognitive meaning of the different network components, showing, for example, this module here at the back of the brain is specialized for perception. That is not a new discovery. That is a rediscovery of one of the basic pieces of neuroscience, that visual cortex is located here and specialized for perception. The more expensive rich club in this network was in fact relatively specialized, shown here in red, was relatively specialized for the higher order, so-called executive tasks that combine both action and cognition. And that gets us to this kind of simple heuristic or working hypothesis, a cartoon I've called it here, of the cognitive connectome. We have a complex topology. Some aspects of that topology, such as its high efficiency, the existence of connector hubs and a rich club, come at higher cost, but may confer shorter conduction times and may favor a more distributed or workspace architecture that's important for domain general higher order cognitive processing, whereas other aspects of the topology, such as the existence of clustering and modules, are cheaper biologically in terms of wiring and metabolic costs, but may constrain greater conduction time and be more suitable for the sort of phrenological architecture, if you will, the locally specialized domain-specific processing of particular cognitive tasks. So in the last few minutes, I'm going to take some of those ideas about the normal human connectome and try and relate them to the disordered connectome. And to do this, we wanted to avoid one of the sort of bear traps of clinical neuroimaging, which is small sample size. It's easy enough to get 10 or 20 patients into a scanner, but then how reproducible are the results likely to be? We addressed this challenge again meta-analytically. So we pulled down data that had already been published using structural MRI to map cortical lesions, if you will, gray matter deficits in the cortex of the brain in about 20,000 patients. And if we simply average those lesion maps over all subjects, we get this result, showing that there are some bits of the brain that are particularly likely to suffer gray matter deficits across a very wide range of cognitive and neuropsychiatric disorders. Interestingly, when you relate the anatomy here to the topology of the normal connectome, it turns out that higher degree nodes have a much higher probability of being pathologically lesioned. It seems that the hubs of the connectome are hotspots for brain disease. Now, we don't know entirely why that is. It could be that hubs are more vulnerable to disease processes, perhaps because they're metabolically more expensive, or it could be that hubs are no more vulnerable than any other part of the connectome, but if they're pathologically damaged, they're more likely to be symptomatic, because we know that they're important in information processing across the system as a whole. In any case, it is true that on average, over all disorders, hubs are disproportionately implicated in brain disorders. And if you look at a particular brain disorder, that story stands up. So here are maps showing the distribution of gray matter damage in two disorders, schizophrenia and Alzheimer's. You can see they largely do not overlap in their anatomical distribution, but in both cases, the probability of a brain area showing gray matter deficit is greater in those nodes of the connectome that have high degree. It's in schizophrenia, the frontal cortical hubs. In Alzheimer's, it's more the temporal cortical hubs. So the hubs that are implicated are not the same across disorders, but across disorders it generally is the case that the brain areas that are most damaged are those that have high centrality in the connectome. Finally, I just want to come back to the question of development. And this is important because when you're talking about many disorders in psychiatry, particularly schizophrenia being one of them, it is a developmental disorder. People typically become or have the diagnosis of schizophrenia for the first time in their early 20s, but there's a lot of epidemiological and other evidence to suggest that their developmental path was not uncomplicated up until that point. So if we want to understand how the connectome becomes abnormal in disorders like schizophrenia, we really need to understand more about how the network normally develops in the human brain. And here we have looked in the 14 to 25-year-old age range using structural MRI, and we've shown, first of all, and this is to replicate some previous findings, that the cortex thins over time. So from 14 to 25, the cortical layer on the outer surface of the brain goes from about 2.8 millimeters thick down to about 2.6 millimeters thick. Over the same period of time, there is increase in myelin, which is the fatty tissue that surrounds axonal projections and enhances their conduction speed. That myelination is coupled to the change in cortical thickness. They're highly correlated, as you can see here. But to the point of the connectome, if you look at where in a structural covariance network derived from these data, the hubs are located. They're shown here, both in terms of degree and closeness. It's clear that the brain regions that show the most rapid adolescent rates of shrinkage and myelination, the brain regions that are most actively consolidated during adolescence, are also the regions that will have high degree in the adult connectome. And finally, we can, again, use genetics to try and understand the implications of this, not just for the normal function of the connectome, but for disease. So here we have mapped the association between the brain areas that show the most rapid rates of adolescent consolidation, shrinkage, and myelination. We found the genes that were strongly associated specifically with those regions. This is an ontology analysis of those genes, and you can see that many of them are specialized for neuronal functions like synaptic signaling, as well as a number of metabolic and ion transport functions. But importantly, this gene set was also significantly enriched for the risk genes for schizophrenia. So the position that emerges from these data, and will need further study in future, of course, is that the risk, the inherited risk for a developmental disorder like schizophrenia may mean that there are changes in local cortical consolidation associated with abnormal formation of network hubs in adulthood. And that abnormal network configuration is what ultimately underlies the emergence of psychiatric symptoms in these patients. So with that, I'll conclude. I've tried to illustrate a number of key points. First, in talking about human brain MRI networks, I've laid out some of the evidence that something as conceptually simple as degree, something as conceptually simple as the existence of a hub in brain networks may turn out to be important. These are biologically costly elements, they're cognitively valuable, they're pathologically vulnerable, or certainly implicated in many diseases, and developmentally consolidated. We need to understand more about how the trade-offs between topology and biological cost play out, and I think we have a fantastic opportunity in this field as it moves into its second decade to get down to an unprecedented level of detail using the new kinds of data from animal models and the leveraging of genetics that I've indicated to understand not just what does the brain connectome look like, but what are its underlying biological mechanisms. So with that, I'll finish, and thank you for your attention.
So thank you very much for a great talk. I think we have time for maybe a couple of questions, just a couple of minutes, then the parallel sessions will start shortly. And I wanted to mention, before I forget, that the map of where the parallel sessions are is in your badge.
Yeah, I have one question. I mean, I completely understood the connectome of C. elegans, because there, you have clearly defined neurons, and you have also the synapses. but I didn't understand at all what your connectomes are on a higher level. You know, so there must be some scaling involved. How many neurons do you have in one of your nodes, for example? And if that is several neurons, then I would like to know, is that scale independent? You know, would you, if you take the same MRI images, is the connectivity changing when you go to another scale? This is really important, I think.
Okay. Okay, well, so the concept of scale and scale invariance is just under the surface of my talk. My intuition would be that if you could drill down from the resolution of an MRI scan in a human brain down to the level of synapses and cells, you would find quite a high degree of conservation of network organization over those scales. That experiment cannot be done for a number of reasons. What can be done already is that you can look across species and you can look across scales and you can look for evidence, at least qualitatively, that some of the network properties that you see at a macro scale in humans are conserved at the mesoscopic scale of the mouse track tracing and at the microscopic synaptic scale of C. elegans. So I think the field is at an intermediate position. We can't do the pure experiment, which is to dive down in the same species from the highest level or scale down to the finer scale. That is not impossible. I mean, that is impossible in humans. But what we can do is we can work across existing data sets at different scales. And as people begin to introduce more and more complete microscopic mapping of some of the larger organisms, Drosophila and so on, it may be possible to link those observations to imaging or interareal connectomes in those species. Yeah.
Okay. Yep.
Okay, and yeah, it was excellent talk. You covered a lot of ground. My question is about the genetic expression. So you told that for the energy consumption, you saw some link, and then, of course, the question may rise, would it be possible to link to learning? I mean, you know, people talk about the possibility of genetic expression. How you see this? So, is that...
Well, so first of all, you can, people have linked network changes to learning in humans. Everything I showed in humans took a rather sort of static view of function, but you can certainly look at how network properties change over time, and if that time period is associated with learning or other behavioral changes, there is evidence for associated changes in network organization. So without getting into genetics, people like Danny Bassett, for example, have already been able to link the dynamics of functional networks to changes in the behavioral learning of those participants. In terms of linking it to genetics, again, we have a situation where, you know, we're somewhat constrained in terms of what can be done in humans. I mean, the linkage of the connectome to human brain gene expression has really only been possible in the last couple of years, and that's because we have now 6 post-mortem brains available to us that map gene expression with the requisite detail. But obviously they're post-mortem. Getting to an association between gene expression and individual differences in cognitive performance like learning and how those 3 things, cognitive performance, gene expression, and network organization might be linked. The only way that I can imagine doing that is if we can find some convenient proxy in humans for brain gene expression that we can sample you know, less invasively than waiting for the participant to die so we can do a post-mortem analysis.
So unfortunately, I've been told that the parallel sessions really do start on time. So I wish we could take more questions, but I don't think we can. Sorry. Thank you. Thanks. Please thank Ed again.